Hello, everyone. This is Dr.Hira, and this discussion is about glycolysis, one of the very important uh, reaction chain, and which is an important part of the cellular respiration. So, in this lecture, we'll talk about the glycolysis steps and what are the key enzymes which are participating in this, and what are the significances of the glycolysis. So, the first thing is, what is glycolysis? So glycolysis is basically the process which that allows the simple sugar, glucose, which comes from your food to be broken down into usable energy, which and that energy is basically responsible for conducting your number of physical activities, including blinking of your eyes, thinking, movement, physical movement. So it's a and inside your body system, there are the number of metabolic reactions that are going to happen, and they need certain kind of energy in the form of ATP. So that will be supplied from uh, these reactions. So it is a part of a large process called the cellular respiration. So you can say this is the key or the starting and one of the part of the cellular respiration. And this cellular respiration include the Krebs cycle, Cori cycle, gluconeogenesis. There are a number of different things. So we are going to discuss the one of that important part from that cellular respiration, which is responsible for the digestion and creating different metabolic molecules for the utilization, for the growth, for the development of our body system, and the energy synthesis. So the glycolysis, in general term, this is the breakdown of the simple sugar glucose, and this is happening in the cytosol. So the cytosol is basically which you term and which are familiar as a cytoplasm. This is the outer watery phase of the cell, right, outside of the nucleus, and the cytoplasm is covered with the cell membrane, and you that you can see from that image. So I have just put that image for your understanding. So you should know where this glycolysis is going to happen. So this glycolysis will happen at the cytosol or the cytoplasm, right? And then they orbit this certain metabolites which then produce, and they will then go into the different organelles of these cells. So the glycolysis term is basically derived from the Greek words the glyco, which is derived for, and it means basically the sweet. So the sugars are largely sweet in taste, so that is then linked with the sugar. And the lysis is basically to break down, so the breaking or the addition of this sugar into the glucose into the further different simpler parts or the components. This is basically the glycolysis. One of the important thing to mention is this one, that the carbohydrates, when we talk about, and the sugar we talk about, so its digestion starts from your mouth. So they are this in your saliva, there are the number of enzymes which just get started when you keep that sugar in your mouth and you can experience the chocolates and some of the candies stuff. So when you keep that in your mouth, so they just turn into um, they melt and they start dissolving, right? So their digestion start from your mouth. Unlike the proteins which just get into the stomach, and then there are the gastric juice, and from the pancreas there are the secretions which then start their digestion. So this is basically which is just starting as you take up in a food, and this is in your mouth, so it just gets started. So that's why it is simplifying into the glucose, and then further the glucose is going to happen through undergoing the further digestion process, and that is the glycolysis. So, what the glycolysis process actually is. So, over the course of several steps, glycolysis break down one glucose molecule into two molecules of another compound that is called the pyruvate. Now, the pyruvate is also called as a half of the glucose molecule, because the pyruvate is basically the three carbon molecule, while the glucose is a six carbon molecule, and you know this is a hexose sugar. So the glycolysis is basically um, you can classify the whole glycolysis chain of reaction into the two phases. There is one which we term as the investment phase, and the other one is the reward phase or the payoff phase, right? So I will tell you that why we say this investment phase and a reward phase. In this slide, you can see that this is the hexose sugar, and this is in a ring structure. And what we call this ring structure, just remember that one, okay? Just refresh that what is a term we use for drawing this a cyclic or the ring structure of the carbohydrate. So I will put some of the questions in your evaluation or under the video. So you just answer me that what this projection call, right? So the glucose is converting into the two molecules of the pyruvate, which are again the three, one, two, three. These are the three carbon structures, right? And they broken up into the two molecules of the pyruvate. Now the glycolysis, it seems that, okay, this is a glucose. Now it is converting into the 2-pyruvate, but this is not as simple as you look in this equation. So there are the 10 steps in this reactions, or you can say the 10 reactions in the glycolysis from 1 to 10. And there is the utilization of the ATP, and there is the synthesis of the ATP molecules, right? But this is glycolysis is not actually the key or the main chain of the reaction to the production of the ATP. So there are further other reactions in the cellular respiration which do involve the ATP production as well, right? So at this moment we are talking about only the glycolysis. So the glycolysis has the 10 steps, and these 10 steps can be classified into the two phases. First to fifth step, termed as investment phase, 6 to 10 step, termed as a reward phase. And why we say this is on the basis of the ATP. ATP is the adenosine triphosphate, and we know that this is a term as the cellular currency. Our body can recognize the energy in the form of this chemical energy, and this is in the form of the ATP. So in the first to fifth step, there is the ATP molecule which is consumed, and because this is involved in the phosphorylation, and this is giving its phosphate group, and there is a production of the ATP. So this energy is consuming, right? So that's why it is called as an investment step. While in a 6 to 10 step, there is the synthesis of the ATP molecules. That's why we call this as a payoff step, because now there is energy which is producing. So now we will go for the significance of the glycolysis product. Like we see that in a glycolysis, there is a 2-pyruvate molecules which will be produced. Now, what is the importance for the pyruvate molecule? So pyruvate is basically which then transform into a very key molecule metabolite, which is the acetyl CoA, right? And this happen in the mitochondria. Firstly, the glycolysis is happening in the cytosol. Then from the cytosol, this pyruvate will get into the mitochondria, and mitochondria, that will be converted into the acetyl CoA. Then that acetyl CoA is basically the starting uh, point or the initial reagent or the molecule which gets into the citric acid cycle. And then there are the electron carriers, NADH and FADH. They are again made in the mitochondria. Oxidative phosphorylation, this process occurs in the mitochondria and uses the electron transport chain to produce ATP, the bulk of usable energy of the for the cell. So the bulk, so that as I have mentioned previously, that glycolysis is not only the key or the main uh, reaction to produce ATP. So there are the further reactions which are happening to produce the bulk of that ATP, which we are using for our body functions. This is the overview, just to mention you the importance or the significance of the pyruvate molecule through the glycolysis. So here you can see that this peach highlight area is basically giving you the idea that these all kind of reactions and the steps are basically involved in the biosynthesis of the molecule. So here is a glycolysis. Glucose is giving you the pyruvate. There is the lactate, which can again convert into the pyruvate, and pyruvate give you the lactate. There are the fatty acids which go through the beta oxidation and they can synthesize the acetyl CoA. Pyruvate can give you the acetyl CoA. Amino acids directly can convert it into the acetyl CoA, and there are the cholesterol, fatty acids, and the ketone bodies, right? So acetol is acetyl CoA then involved in the production of these one, because these are also the very important ingredient for our proper body function and the growth. So this is basically involved in the synthesis of the molecules, while this grain area is basically involved where this acetyl CoA is the part of the cyclic reaction steps, right? This metabolism. So this is called as ATP synthesis, because there is the bulk of that ATP which will be produced. So this ATP production is basically the aerobic respiration we term as this, and then all of this glycolysis is basically under the anaerobic condition, because when there was evolution of this life on earth, so that was starting with the absence of oxygen. So the glycolysis has developed in a way that this is not requiring that oxygen to convert that glucose into the pyruvate one, and that is basically the blessing or the initial evolution step, that why the glycolysis is basically the anaerobic kind of reaction. So here, now we are going to start one by one. That what are the key 10 steps of the glycolysis? And you have to see that in each step, what kind of structural or the chemical changes are happening with the glucose molecule. What are the different end product in each step? What are the specific enzymes which are involved? So the very first step is basically the phosphorylation of the glucose. So we are adding up the PO3 group to that glucose molecule. So this is basically to generate the glucose 6-phosphate, another hexose molecule, but there this hydrogen is replaced with this phosphate group, right? And the enzyme which is involved here, this is the hexokinase, because this is working with the hexose sugar, and the kinase is basically involved with the synthesis. So there is a phosphorylation which is happening, and this modification is happening with the glucose, and now this is a glucose 6-phosphate. Now another thing is obviously to convert the glucose to the glucose 6-phosphate, so we need some of the source which is providing us the phosphate group. So that is the ATP, adenosine triphosphate. So this is donating one phosphate group to the glucose, and then we have this glucose 6-phosphate, and there is ADP, which is adenosine diphosphate, and there is the hydrogen ion which is produced, right? Another important thing is this one, that this reaction is uh, also required the magnesium ion as a coenzyme or the cofactor to happen this reaction. So here you can also realize the importance of some of the metals in your body, that they may not require in the high amount, but even in traces and a minor amount, this is very essential. So if there is a magnesium is not there, so this hexokinase is not going to happen, not able to perform its this function properly. All right, so this cofactor is very much important here. So this is the importance of these metal ions in your biological system. So the next step is now from the first step, if you're taking up that glucose 6-phosphate, right? So what is happening in the second step? The second step is basically the rearrangement of this glucose 6-phosphate into the fructose 6 phosphate, right? And eventually we will get the fructose 1,6 bisphosphate. So this is a kind of reaction where there is an attachment of another phosphate group, and there is the rearrangement of the molecule. So here we can see carefully and what is happening here. So this is a phosphate group, right? The first thing which has happened to this one is that this has rearranged into the fructose. Now there is a question that why it get in converted into the fructose, why it wasn't happy with the glucose 6 phosphate, because it need to convert into the two sides with the phosphate group, right? So there was a need that there should be another alcoholic carbon which should be free, and obviously then there is the happening that this hydrogen can be replaced with the phosphate group. So that's why this glucose 6 phosphate has take that step, and this has rearranged itself in the presence of the phosphoglucose isomerase, right? Because now this is the just a rearrangement, this is converting into in its one of the isomer. So we get the fructose 6 phosphate. Further, there's another involvement of the ATP, and again, phosphofructokinase enzyme is there in the presence of the magnesium, and this another second phosphate group get attached to this one, right? So the first was with the 6 carbon, another one with the first carbon. So the two extreme carbon, which are the free, or you can say the primary alcoholic structure in the carbohydrate or in the glucose one, and then in the fructose one, so they get phosphorylated, right? They get attached with the extra phosphate group. So obviously there is a ATP. So up till now to the second step, we see that there is the investment of the two ATP molecules. So just count that also with each of steps, so you can at the end you will be able to count that how many ATPs has consumed and how many ATPs has synthesized, and what was the overall um, um, generation or the output in terms of the ATP in the case of the glycolysis. Now moving to the third step and the fourth step simultaneously, so the N enzyme comes along and cuts glucose 6 phosphate into two, three carbon molecules. The enzyme are starting to look a lot more like two pyruvates, right? The step 4 is the enzyme aldolase split fructose 1,6 bisphosphate into two sugars that are isomer of each other. These two sugars are dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. So here you can see, so the three step is basically you can see that you can merge into what has happened in the first step, but these are usually mentioned as separately because then it was converted into the glucose 6 phosphate into the pyruvate one, right? So here you can see the first thing what is happening is the fructose 1,6 bisphosphate. These are, bis is for the diphosphate, you can say, right? And the aldolase enzyme is responsible for converting it into the two isomers, and these are basically the glyceraldehyde 3 phosphate. It is called the short abbreviation is a GAP, and the DHAP, dihydroxyacetone phosphate. So now you just have to tell me that what is the difference between these two isomers, right? So what kind of carbohydrates they are, and in a generic class of carbohydrates, how you can place that one. So glyceraldehyde 3 phosphate is basically the aldose sugar, and this is basically the ketone kind of sugar which is producing, and each of this is carrying one of the phosphate group. So triphosphate isomerase, this is an enzyme which is converting this dihydroxyacetone phosphate into uh, this one, right? So this is happening, and there is a break off, or you can say the cleavage of this fructose 1,6 bisphosphate into the three carbon structure two molecules, right? So here you can just correct this one rather than if that is confusing, because this is just taking up with the previous one. This is linking with the previous glucose 6 phosphate, but you can write it because there are this fructose 1,6 bisphosphate, which is converting into these three carbon structures. Now, the fifth step is basically the enzyme triphosphate isomerase rapidly interconvert the molecule dihydroxy acetone phosphate and glyceraldehyde 3 phosphate. Glyceraldehyde phosphate is removed, used in the next step of glycolysis. This is the end of the investment phase, right? So it will be consumed and removed, right? And that you can't see further. So this dihydroxyacetone phosphate will be further uh, utilized in the form of glyceraldehyde 3 phosphate, and that is basically responsible to moving into the further reactions of the glycolysis one. So here from the first to the fifth, this is the investment phase where we were utilizing the ATP. And do remember, you have noticed that till now, we have consumed the two ATP molecules for that glycolysis steps. Now, the next is in the reward phase, ATP and NADH are made. This is the payoff of the original molecule of ATP invested in the first step. So the sixth step started with the glyceraldehyde 3 phosphate gap, as I have mentioned. This is a key, basically, three carbon structure, which firstly formed from the fructose 1,6 bisphosphate, right? And the other is a ketonic form just converted into this aldehydic form, because this is the most stable one and the aldolase enzyme is there. So we get that gap as a key molecule, and then it will convert into the further metabolite. So the glyceraldehyde 3 phosphate with the NAD plus inorganic phosphate, right? And this is the GADPH, which is a molecule, right? This is a glyceraldehyde phosphate dihydrogenase enzyme. So this is converting into the 1,3 bisphosphoglycerate, right? And this here you can see this is the hydrogen which has removed, right? And there is the hydrogen um, which has removed with the phosphate group. So basically, this is the phosphate group which we initially get here. This is a third carbon where we have initially the phosphate group, and then the first carbon, which is the aldehydic, the anomeric carbon hydrogen, that is again replaced with the phosphate group, right? And this is the phosphorylation. So this is the O and the pH, right? So it means this is the kind of oxidation also which it get happen with this one. And there is the NADH which get reduced from NAD to the NADH, and there is a hydrogen ion again forming as like the ATP production we have seen in the previous slides. Now the seventh step is the ATP is formed as a phosphate group is removed from the three carbon molecule and added it to the ADP. So here 1,3-bisphosphoglycerate, this is giving the one phosphate group to the ADP, and we will get that one. This is the 3-phosphoglycerate, and there is a production of the ATP. So here from this we are starting the synthesis of the ATP. So this is the first ATP molecule which is formed in the glycolysis. So here you can see this is the phosphoglycerate kinase enzyme, and again this is the magnesium one, and this is the production of the ATP one. Now the eighth step is the phosphate group shift from the third carbon to the second carbon. So this is basically the rearrangement, right? So the enzyme which is responsible for carrying out this activity is basically the mutase enzyme. So phosphoglycerate mutase, because this is just rearranging or the shifting this phosphate group from third carbon to the second carbon. Now, the ninth step is basically the enzyme enolase removes a molecule of water from the 2-phosphoglycerate to form phosphoenol pyruvic acid. This is PEP. This is called the PEP. So the phosphoglycerate you have seen there. 8 is basically when we have the ester kind of ester functionality. There's a carbonyl and there is O, but this is not having the hydrogen one, right? So acetate, this is just extract of the acetate one because this is the ester formation here. So the enolase enzyme is converting this one into the one of the unsaturated structure where there is a double bond between this carbon and this carbon, and there is the removal of this water molecule. So let me highlight this one, that what has actually happened to this one, that by this arrangement this has favored that there would be the removal of this water molecule. So that water molecule removal is enabling the formation of this PEP. This is a phosphoenol pyruvate. Now moving towards the next one, this phosphoenol pyruvate, by uh, giving its phosphate group to the ADP in the presence of the pyruvate kinase enzyme again. So here you can see this is converting into the pyruvate molecule, and there is the production of the ATP. So this is the second ATP molecule which we get in the step 10, right? So the enzyme pyruvate kinase transfer a P from phosphoenol pyruvate to ADP to form pyruvic acid, and ATP result in step 3. The second ATP is created as a phosphate group leaves the third carbon molecule and is added to the ADP. Pyruvate has been made. So finally, you have a pyruvate, right? So by looking at this one, what you have confirm here, that there is for the each as a glucose is breaking into the two molecules of the three carbon structures, right? So there is a one pyruvate we have discussed is, so the two ATPs have produced from this one, and two you should keep in for the second pyruvate and molecule. So it means in a total there are the four ATP which produced in the glycolysis. Now we have to calculate overall what was the total we get and we have invested in the glycolysis. So during the glycolysis a total of four ATP are made. Recall that the original six carbon molecule is split into two three carbon molecule at the end of the investment phase. This means that the two three carbon molecule both go through the reward phase, both yielding 2 ATP. Remember, 2 ATP were used in the investment phase. So we have a net production of 2 ATP molecule, right? So to summarize for more emphasis, I've just listed in a points that there is the minus 2 ATP means that they have consumed in the investment phase, plus 2 ATP reward phase for first pyruvate, plus 2 ATP plus is a sign for the production for release. So in a reward phase for the second pyruvate, additionally, there are the two NADH compounds, two water molecule, and two pyruvate are created. So that is a net result of the glycolysis. So in the last slide, I'm just giving you the whole of our overview, what we have going through and we have discussed in the glycolysis one. So just a review. And if you want to summarize the whole of the steps in the single sheet or in a single flow or the blocked flow diagram. So you can see that when we started with the glucose, right? And they are the delta G values for the each step. Here you can see this is a minus because this is utilizing the ATP. Then there is the positive delta G. Then there is again in a minus value. Then we have these three carbon structure molecules. They going through these steps. These are taking up all these delta G, which is a free energy, and the positive sign means this was the hexagonic, endergonic, and these are the exergonic reactions, right? So here there's a production of the ATP, which is energy producing. Then there is a production of the ATP. So overall, we get the 2 ATP molecule production for the one single glucose molecule. So that is the nutshell of the glycolysis, covering all of its steps. So I hope that give you the insight of the glycolysis and what are the importance of this one, and what are the key steps, what kind of changes are happening with the glucose step by step. So if you still have any questions, you just can comment or you can write to me. So I would be happy to answer your queries. Thank you so much. Have a good day.